All NEET aspirants, you are listening to podcast Botany for NEET by S. Ramakrishna. In this podcast, packaging of DNA helix. It is one of the tricky topic in case of molecular basis of inheritance because we can connect the dots to various corners of the molecular basis of inheritance like gene regulation, gene expression, transcription, translation, transcription machinery, how these words are related to the packaging. So, connecting to the dots is the duty of the lecturer, and conceiving to connections by listening with attentivity is the duty of the student. So, listen with attention. Whatever the question is going to be tricky or a trap question. First you have to know about why packaging is essential. What is packaging? In case of DNA packaging, why packaging is essential? Because the DNA is a long molecule. Nucleus is with small dimension. So long DNA molecule accommodated in a small dimension nucleus possible by means of compactation packaging only. So packaging requires certain materials, the packaging materials like histone protein and non-histone protein. Next, the packaging, that is packaging of the DNA for protection. Not only pack for protection, unpack for expression is also important because the genes of the DNA are all not expressive in all occasion. So the packaging ensures the accessibility of the genes to be expressive in a required occasion. So packaging is meant for regulation of the gene expression. So packaging is flexible and reversible. How it is flexible and reversible? A dynamic process because it is an electrostatic interaction. It is not a covalent interaction. How can you say it is an electrostatic interaction? DNA it is acidic due to phosphate and histone protein which are basic because of abundance of lysine and arginine. So it is an electrostatic interaction, reversible, flexible and dynamic. It is not a question of the genes. It is the accessibility of the genes for expression is the duty of the packaging of DNA. Next, how can you calculate the length of the DNA? Distance between consecutive base pair into total number of base pair. Distance between consecutive base pair 0.34 into 10 to the power of minus 9 meters. If you calculate the mammalian cell, the DNA diploid content, length of the DNA is equal to 0.34 into 10 to the power of minus 9 into 6.6 into 10 to the power of 9 base pair. It gives 2.2 meter long DNA. The 2.2 meter long DNA accommodated in a typical nucleus of 10 to the power of minus 6 meter. How it is possible? By means of packaging, compactation only. Previously a NEET question in this. In 23 chromosomes, how much length of DNA accommodated? That is 1 meter or 1.1 meter. In 46 chromosome, 2 meter or 2.2 meter DNA accommodated. Next, in NCERT there are the two lines with regard to the Escherichia coli. If length of the DNA is 1.36 millimeter, calculate the number of base pair. 10 lakhs nanometer is equal to 1 millimeter. If you calculate this, approximately it is going to give 4 million base pairs. Next, packaging in case of the prokaryote. Even though prokaryotic bacteria, which is not having true nucleus, that is without nuclear membrane, DNA is not scattered. Why? DNA held with a non-histone protein as a large lobes in nucleoid. So DNA is looped in case of prokaryote. So packaging is very simple, and in this also it is reversible because it should be accessible for replication as well as transcription. Next, DNA packaging in case of eukaryote is much complex than prokaryote because packaging occurs at various levels and various types of proteins are required at various level. That's why it is complex. Next, for packaging, the histone proteins initially required. The histone protein or basic protein positively charged because of abundance of lysine and arginine amino acid. Not with all arginine and lysine, abundance of lysine and arginine. Lysine and arginine acquire the positive charge because of the side chain, that is the R group. Next, the organized histones forms a spool or a ball. What do you call it is histone octamer. The octamer word itself conveying its meaning. It is 8 histone, H2A, H2B, H3, H4, each having 2 copies. So it is histone octamer. Negatively charged DNA wrapped around the histone octamer sealed by H1 protein that constitute a nucleosome. So basic unit of DNA packaging, what do you call it is nucleosome. So one nucleosome having 5 types of histone and 200 base pairs of DNA. And this nucleosome, it is a repeating units in a structure in a nucleus. Repeating units in a structure. What is the structure? Chromatin in a nucleus. Threaded bodies, that is colored threaded bodies in case of nucleus. So nucleosome appears as a beads on a string. Beads are nothing but nucleosome. String is nothing but chromatin. So each bead is equivalent to, that is nothing but nucleosome with a 200 base pair. Theoretically, it is possible to calculate the number of such beads in case of mammalian cell. If we take the diploid content, 6.6 into 10 to the power of 9 base pair. As 200 base pair gives, that is equivalent to 1 bead. 6.6 10 to the power of 9 base pair is equivalent to how many beads? Roughly it comes under 3.3 into 10 to the power of 7 beads. Suppose in case of equally 4.6 into 10 to the power of 6 base pair, how many such beads are possible? In this beads are not possible because there is no nucleosome concept in case of prokaryote. Next, beads on a string of chromatin packed into chromatin fiber. The chromatin fiber further condenses by utilizing non-histone chromosomal protein at a metaphase to form chromosome. These non-histone proteins, which are scaffold protein, which provide the support to give a particular structure. So particular structure maintaining finally is important. Such scaffold protein, that is non-histone chromosomal protein, are utilized at higher level of packaging in the formation of chromosomes. Next, the chromatin loosely packed, take less stain, light in color, transcriptionally active, what do you call euchromatin, because the regions which are accessible to transcription unit, because it is loosely arranged. And chromatin tightly packed takes much stain and dark in color, not accessible for transcription machinery. It is inactive, what we call heterochromatin. So euchromatin transcriptionally active, heterochromatin transcriptionally inactive, and accessible to transcription machinery euchromatin, not accessible to transcription machinery, it is heterochromatin. Such differential stainability because of what? Because of coiling nature, loosely coiled and tightly coiled. So in this, if you frame question, what is the basic unit of DNA packaging? Nucleosome. Each nucleosome having how many types of histone? 5 types of histone. H2A, H2B, H3, H4, H1. So, how many types of histones are present in the core of nucleosome? Four types: H2A, H2B, H3, H4. And extra nucleosomal histone is, that is H1 protein, like this. So, I am signing off from this episode. You will see in case of the next episode the search for genetic material and properties of genetic material.